recorded participants, we expected about 25, and I think it was 178. And more importantly, we had 40 researchers who asked to speak on subsequent webinars. So we created a webinar series at that moment and said, welcome to the community. And so since then, we've done, I think, about 26 of these, Lauren. You know the numbers better than I. And very interestingly, we've had over 11,000 views of the webinars and the individual researcher PI lightning talks, which is really kind of incredible. It was funded initially in 2020 by NSF, and then with an extension in 2021, a $2 million extension to include NIH awards. You know, we said, you know, we're very grateful that NSF has funded this, but this is a disease and we really feel like we should be working with NIH too. And so we're very grateful that we got this 4-year extension through 2025. And since then, we initially had 732 NSF COVID-related awards. Now we have over 10,000 NSF and NIH COVID-related awards. And interestingly, if you think about it, maybe at the beginning you would think, well, it probably says SARS-CoV-2 or coronavirus in it, but there are many other implications, you know, and societal impacts. So it could be, you know, education preparedness, it could be child rearing, it could be transportation challenge. There's all sorts of things that COVID has impacted. So it's broader, or mRNA, you know, we maybe weren't thinking about that initially unless we were the biologists and pharmacists working on that. So it actually has a wide range of research that's related in some way to COVID. So it's a portal and a community, and we're delighted that you could all be part of it. If you go into the COVID Information Commons website, you'll see a COVID Awards and PI, or Principal Investigator database. If you click on that, you could put your name in or someone else's name in, see if you have a relative that got an award, and it will come up with the NSF and NIH awards that are in the database. You can also look by NSF Directorate, NIH Institute or Center like NIAID, or any of the others. Or awardee organization, state or territory. Most of the awards are in the United States. There are a few outside the US from NIH, but it's mostly US. And so you can actually easily find the award information. You can actually click on the name of the PI, and if they've given us additional information, we have PI profiles. So like if you put in Hudson and you clicked on my name, you would go to my profile, and my ORCID ID is there, a video, my lightning talk video is there, keywords about the research, links to the websites. And so any PI can actually do that as well. So it's a real opportunity to share and make it more fair, findable, accessible, interoperable, and reusable. There's another tool, which is our machine learning map tool, which is also available from the homepage. And you can see that we have 10,200 approximately awards now. It gives you this kind of like topical view of mapping, clustered by topic using machine learning. And you can also look at the next slide, and then this would be the faceted search that is built on Lingo4G, which is a machine learning clustering tool from a company called Carrot Search out of Poland. And you can put in a specific keyword like epidemiology. It'll, you know, chunk it down to 367 documents in scope, not 10,000. And you can see it did it in 389 milliseconds. So it's pretty, so if someone's been using it, it's pretty fast. And then you can click on any of the awards, and then you can see the abstract on the right. You can see that this view shows a clustering of dynamics, transmission, epidemiological. Those are some of the keywords. Let's say University of Florida, you're like, gosh, I wonder who's there. If you click on that, and then you click on the little gears, which I call the Mickey Mouse head in the top right, it'll actually show you the PI name. So we make this very accessible. It's been used by students and by researchers to find other people in their topical research areas. And this is always open and live for everyone to use.
Yes. Thank you so much, Florence. Another program that we actually want to highlight in today's talk is our 2023 KIC Student Paper Challenge. And earlier this month, we announced the winner challenge. We had 2 cohorts, as you can see, some graduate students and undergraduates. And the students addressed a range of topics in their papers, including COVID and diabetes, data clustering, social media disinformation, COVID diagnostics, and then quarantine policies. So we're so proud of all of our students. I think a few of them are actually here today with us in the webinar. So we're so thrilled that you could join us today. Congratulations again to this year's winners. And to all of you, please keep an eye out for an announcement in 2024 for our next KIC Student Paper Challenge for this upcoming year. And one thing that I do want to make sure to announce as well for the other students in our webinar today is the KIC Student Working Group. We actually have two working groups for the COVID Information Commons. This one is spearheaded actually by two of our work study students at Columbia University. This KIC Student Working Group currently has over 160 members. We meet monthly. The members are from all over the world in different academic institutions and organizations. It's a really great space for students who are interested in working together on data science projects that are related to COVID. And actually this semester, we are trying something a bit new. Just yesterday, we launched the Fall 2023 Data Visualization Challenge. So we welcome anyone who's interested in data science, whether you are new or super experienced, to try out the visualization challenge that our student leaders have created. This will include materials that show you how to take a data set and transform it into a working visualization. And in our monthly meetings and office hour sessions, the students can workshop their work together, get peer feedback, and improve their skills. This is meant to be a very low stress, friendly learning environment. We encourage everyone to join. And the next time that we will be meeting as a group is Friday, October 26th at 10 a.m. So we hope that you or some of your students will join us in that session. And you can find more information about this program on our website. And I will be dropping the links to that in the chat in a minute. And we also have, as I mentioned, our second KIC research working group. And this is more oriented towards researchers and faculty members. And so our members here are from 58 different organizations across 32 US states and territories, 3 other countries. So again, this is a really collaborative and international space. We hope you'll consider joining us for our next meeting, which is tomorrow, believe it or not, Wednesday, October 11th at 3 p.m. To receive a calendar invite for this meeting, you can just ping me directly. I will be dropping information about this in the chat as well. So all in all, you can see that we have a lot of fantastic projects happening at the KIC, and we're so excited to offer all these different collaboration opportunities to you as researchers in this space. But without further ado, I want to hand you over to our main speakers for this afternoon. I'm going to introduce our speakers for today's KIC webinar. We actually have 4 fantastic speakers. Some of them have actually presented with us previously at KIC webinars, so we get a bit of an update on their research, which is really fantastic. And if you have any questions for our speakers during their presentations today, please feel free to drop your questions directly into the chat, or you can hang on to them for our Q&A session at the conclusion of these 4 presentations. But I will kick off today's afternoon webinar by introducing you to our first speaker, Dr. Bix, a professional clinical translational research and the director of the Neuroscience Research Center at Tulane, and he will be sharing with us his research on the impact of COVID-19. So let me stop sharing, and I'll hand it over to Gregory.
All right, thank you very much. See if I can do that. Can you all see my screen? How about now?
Yes.
Good to go? You
Yes,
can't see it?
great. Yes, we can see it. It looks great.
Good. Okay, great. All right. Well, thank you, Lauren. I really appreciate that. All right. Well, today I'm going to talk to you about some work that I've been doing over the last couple of years since the pandemic started, looking at novel interactions that the virus has with receptors that we've identified in the body, and then how we can therapeutically target that and the impact that that has on the brain. So our work is really focused on the spike protein. I'm not sure if can you see my pointer as I move it on the screen? Okay, excellent. So our work is really focused on the spike protein, which the virus uses to bind to cell surface receptors and ultimately invade cells. And we identified pretty early on in the pandemic, we and others, that the amino acid sequence of the spike protein contained a particular 3 amino acid structure called RGD. This is a particular binding motif in protein structures that recognizes a particular class of cell surface receptors called integrins. And what was particularly interesting about this, twofold. One, that SARS-CoV-2 was relatively unique amongst other coronaviruses that you can see listed on the screen, in that it had this binding motif where these others did not. And it was speculated pretty early on in the pandemic, in early 2020, that the presence of this integrin binding domain on coronavirus spike protein might convey some of the virulence that the virus has unleashed upon all of us. So with that being said, we and others looked at what was happening to this particular spike protein as the various variants emerged over the last couple of years. And sure enough, with each mutation in the spike protein or mutations, this integrin binding motif, this RGD, which was already fairly prominently expressed at the tip of the spear, so to speak, was even more prominent with various mutations. And here you're looking at the UK variant, but subsequent variants have borne this out. So there is a specific receptor binding domain on the tip of the spike protein of coronavirus that conveyed a unique ability to bind to this class of receptors known as integrins. All right. So integrins, very basically, they are composed of 2 subunits, an alpha and a beta, and they bind to something called extracellular matrix. Extracellular matrix is essentially the glue that helps hold cells together. It forms basement membranes. Every cell in the body expresses integrins of different complements. These subunits are referred to as alpha and beta, depending upon which alpha binds to which beta or associates with which beta. You get receptors that have different affinities for different components of the extracellular matrix. We were particularly interested in some that are expressed in the brain. And this schematic essentially shows integrin subunits that help form what's called the blood-brain barrier. I'll be getting back to this in a little bit. But essentially, there's a barrier that isolates blood and vascular compartment from the brain and the brain parenchyma. And this is tightly regulated. And viruses and other infectious agents can impact the blood-brain barrier. And as a result, there can be infection in the brain or swelling, inflammation, that sort of thing. But these integrins that I was referring to help regulate the tightness of this blood-brain barrier, which will become very important in a minute. And then we've spent a lot of time in my lab looking at specific modulators or inhibitors of integrins. And then one that I'm going to talk about a fair bit, something called ATN-161. This is a peptide that can bind to integrins such as alpha-5 beta-1, which we have since identified as a COVID receptor. And we've done a lot of work to look at whether or not this inhibitor peptide might actually have therapeutic efficacy against coronavirus. Right. So this is a summary of a couple of years of work, but suffice it to say, here you're looking at the spike protein of the virus. It interacts with known receptors. ACE2 is one that you've probably heard of, and associated integrins. And this interaction facilitates the virus getting taken up into a cell, and then all the bad things that happen as a result of that post-cell invasion. So we've done a lot of work to demonstrate essentially that if one takes just the spike protein and puts it on cells in culture, in this case, these are brain endothelial cells, we looked at a number of different indicators of blood-brain barrier integrity, and we see that the spike protein by itself can actually change the expression patterns of these proteins associated with blood-brain barrier. It reduces them, which is a clue, and since validated by others, that the virus can actually impact vascular barriers. We did a lot of work early on in the pandemic to essentially show that these spike proteins can bind to integrins, and that we could block this interaction by co-incubating with this peptide I referred to before, ATN-161. This was ELISA studies that were done pretty early on in COVID-19. We showed, I think equally as importantly, that if we added cells, the virus rather, live virus to cells, we could block a lot of the viral replication by adding this inhibitor to alpha-5 beta-1 integrin. That's shown by graph here. And here you're just looking at the cells that were co-incubated with virus. All these little brown, round balls or plaques are cells that are impacted by the virus. When we treat it with our ATN peptide, we can largely suppress these round cell formation, which is synonymous with viral replication. So in animal models, there are a number of different animal models for looking at COVID-19. My lab's taken advantage of a number of different animal models as they become available. In one particular model, you can make a normal lab mouse express the human form of ACE2, which is a known receptor for the virus. At the time, this was the only way to make a standard lab mouse susceptible to infection. If we inoculated these mice with coronavirus and then treated them with ATN-161, we could significantly suppress viral load in the animals. And in a different model where instead of making the mice express the human ACE2 with the virus, we could actually use a transgenic mouse, something called K18, that expressed human ACE2 everywhere. Similarly, if we infected those mice, we could actually block the infection by treating with ATN-161 as well. But a better tool that emerged in the last year or so is a version of coronavirus that was mouse adapted. So this was a SARS-CoV-2 that was passaged in standard laboratory mice, put into a mouse, allowed to infect, taken out of the mouse, passaged into a different animal, et cetera, et cetera. After 10 passages, the virus could actually infect wild-type mice. This really facilitates coronavirus research in labs because now one can work with standard mice that most labs have access to. And so we took advantage of that. And we did a number of studies. And when we infected mice, just and waited 2 or 3 days and started looking at markers in the brain, we noted that those same tight junction proteins, these are proteins that stabilize, facilitate the tightness of the blood-brain barrier. Here you're looking at claudin 5 is one. We see a significant reduction in the levels of this protein. Again, that's important to blood-brain barrier stability. We also see a significant increase in inflammatory cells and astrocytes in the brain with coronavirus infection. Now, I should point out the virus is put into the lungs. So we're looking in the brain 3 days later. And this is in the absence of virus actually getting into the brain. So we're seeing changes in the brain that indicate blood-brain barrier disruption and neuroinflammation 3 days after infection. This is just another example. We're looking at a stain from microglia, which are the brain's resident immune cells. There's a significant increase in immunoreactivity of microglia 3 days after coronavirus infection in standard mice. So this is just another schematic, again, where we infected mice, standard lab mice with MA10. And then we also treated with that ATN-161. And very nicely, we saw effects on various markers of inflammation. This is interleukin-6, pro-inflammatory cytokine in the brain. We saw that ATN-161 could significantly reduce the pro-inflammatory cytokine in the brain. We saw similar effects on blocking the reduction in claudin 5, which again is a marker of blood-brain barrier integrity. Very briefly, I think everyone on this call is familiar with long COVID, so I won't belabor this slide. But suffice it to say, one of the changes that have been noted in clinical long COVID is a reduction in blood flow to the brain. And there's a number of different reasons why this may occur. But we decided to model this in those standard laboratory mice by putting coils on the carotid arteries. These are major blood vessels that feed blood to the brain. And if you restrict but don't eliminate the blood flow, typically what happens in a standard mouse is after a couple of weeks, there's white matter injury in the brain, And after about a month, these animals begin to develop cognitive impairment. So it's a chronic hypoperfusion of blood to the brain, resulting in a cognitive decline, a so-called vascular dementia. We did a number of studies that essentially showed that if you do this, wait about a week after you put these coils on and then infect the mice, their brains are much worse than if you just infected them alone or if you just put the coils on by themselves. This is alpha-5 integrin, which is a receptor we'd identified for coronavirus. Levels in the brain go through the roof. We saw changes in microglia. We saw pretty significant changes in GFAP, the astrocyte marker, again, in the brain. I'm going to skip ahead. Essentially, if one looks months later after a infection in laboratory mice, we see clear indications of neuroinflammation that are chronic. So this is 30 days post-infection. We see changes around blood vessels in the brain that persist, an experimental long COVID. And we see significant dropout of neurons in parts of the brain where you don't want neurons to go away. In this case, the hippocampus, which is important for the consolidation of new memory into long-term memory. This is 60 days after infection. And a number of the mice, we saw significant neuronal cell dropout. And this is just showing signs of inflammation again that were chronic 2 months after infection. So I want to leave you with this idea that we're now at the stage where we can nicely model coronavirus infection in standard laboratory mice. And unfortunately, in a number of these animals, days to weeks to months after infection, we see changes in the brain, which are made worse when we superimpose reduced blood flow, when we model cognitive decline. And however, we think there are particular receptors that make coronavirus particularly virulent that we could also modulate, in this case with ATN-161, which we are also studying as a potential dementia therapy. And I think because this receptor may play a role in both dementia and vascular integrity and coronavirus infection, it may serve a dual purpose in both treating the virus as well as the vascular sequelae associated with it. So I think I hopefully haven't gone too far over. I'll just acknowledge the team that helped do the research and my collaborators, and I guess we'll wait for the very end to do questions, but thank you very much for your attention. And with that, Lori, can you unshare my screen?
Yes, I can unshare your screen, although I'm reluctant to because it was so interesting. But thank you very much for sharing your presentation with us.
My pleasure. Thank you.
Thank you. And I'm now going to actually hand us over to our next speaker, Zachary Boyd, who is an assistant professor of mathematics at Brigham Young. And Dr. Boyd previously presented his research on COVID and the global supply chain in, I believe it was April of last year. So obviously a lot has changed since we last heard from him. And Dr. Boyd, we're very excited to hear about your research. Thank you.
Thank you, Lauren. Yeah, I'm excited to be back here and talking again. I think this presentation is almost the opposite of the previous presentation. We don't have anything wet or gooey in what I'm doing. I'm a math professor, and I'm focusing basically on understanding the mechanisms of supply chain breakdown and supply chain reorganization, which obviously is really relevant to COVID as well as other disasters. I'm presenting joint work with several people. This is a multidisciplinary team. Nitin is a business professor. Sean and David are computer scientists, and Tyler Burrows is the PhD student who really exercised a bunch of leadership on this project. So huge credit to him for this work. But I'm going to present the results of a mathematical model. It's an agent-based model, which in the realm of mathematical models tends to be very detailed. You can bake in all of the assumptions that you can think to make and really trace out the consequences of those assumptions throughout the system. And the take-home message is going to be basically about the role of managerial decision-making in times of crisis versus in times of stability, and how to think about the trade-off between different policy options within firms. So to start off, just to summarize a little bit of what I already said. So this is basically all based on a novel, detailed model of supply chain, and it is different from a lot of other models which really assume that firms are hooked in a serial manner one to another up through the global economy. We know this is a really unrealistic assumption. The global supply chain really forms this complex web of interactions, and often it even has cycles with firms eventually relying on the products they themselves produce far down the stream in complex ways. So we thought it was important to introduce possibilities of more complex structures to the supply chain than just a chain. We also have included various complex dynamics and nonlinearities, especially accounting for the way that managers might think about things. The managers in this model are mostly risk minimizers. They really want to deliver on their promises to other companies and to their superiors, and they want to choose suppliers and order from them in a way that really guarantees their output to be reliable. And it pulls in a lot of big tools from engineering traditions and computer science and mathematics. And yeah, so some of the big themes you're going to see in the next few minutes is the relationship between the structure of the relationship between firms and the behavior of those firms, and especially the role of policy. Here, I don't really mean public policy, which is kind of what a lot of COVID talks about in terms of policy. This is firm policy. This is how firms act and the way they make their decisions. And we demonstrated, at least within the assumptions of this model, that diversifying the supplier base, which is a big theme for firms in recent years, and especially since the pandemic, firms are thinking about more ways to diversify the suppliers that they source from to try and de-risk themselves. But we've shown that there are both risk reducing and risk increasing effects to these decisions that firm makes, and that the policies firms choose can affect this outcome. So this work is forthcoming in one of the major computer science and control theory conferences, and patent pending. Just to give you a schematic, I'm not going to ask you to understand the guts of a large mathematical model here. That's not the point. But just to give you an idea of kind of what's going on under the hood, we have here a graph of a supply chain. So this is just a tiny fragment of one medical provider that might need medical supplies from more than one firm. And we treat the firm as input and as having inputs and outputs. We know that firms have outputs, right? They have to produce physical goods in many cases and pass them downstream in the supply chain, but there's also information flowing upstream. And that's the other important channel here, because information is unreliable and uncertain and often delayed in times of crisis as well as in times of stability. So we have information flowing backwards up the supply chain about what is wanted in the future. Firms have to make orders and make contracts and push physical goods downstream. And one of the main negative behaviors that we focus on is what's called the bullwhip effect, which is this phenomenon that even in times of stability, when user demand for products is fairly stable downstream, little fluctuations in demand get amplified the further upstream you go in the supply chain. So there's two parts of this. First, material orders tend to be more variable than demand signals. There's a few reasons for this. It's actually kind of contentious what the most important reasons are. But basically, firms may want to hedge their bets and maintain inventory. They're uncertain about the future. They want to really make sure they can deliver on their promises. But then when the second firm in line has to hedge on their bet, then the second firm has to hedge on that hedged bet, and it goes upstream and kind of gets worse and worse. The uncertainty magnifies. And this can be really disruptive. This is why, I mean, I've certainly heard people say that semiconductors are like this, where it can be very feast or famine. When you anticipate that there will be a need, then production has to ramp up like crazy, but then overproduction can sometimes happen, and it's just really disastrous. So we're investigating the role of policy in this context. So there's two, and really managerial control and adaptability. So the main thing I'm going to show you in this presentation is whether the manager takes the stance of being very flexible about who they order from in times of uncertainty, or whether they just continue business as usual. And of course, there's a whole spectrum of how adaptive people can be. But basically, we pin down that in times of stability, managers who fit the assumptions that we've integrated from the literature here basically tend to make matters worse in terms of the bullwhip effect by over-adapting to noise coming from downstream that they actually interpret as signal. But as I'll show you in a coming slide, in times of crisis, the picture is really different. But basically, for the purposes of this presentation, we're thinking of two policies. One, where a firm has, say, 4 different suppliers of a critical ingredient, And they just, maybe due to long-term contractual requirements or company policies or just managerial decision-making, they always source the same proportion of what they need from all their suppliers and hope that their suppliers come through. Whereas on the right, if you have a lot more managerial liberty, or you believe that you might be entering a time of crisis, then there's this variable ordering policy where the manager has to use past behavior and forecasts of the future to guess which of their suppliers are going to come through for them and how much they're going to come through, and adapt their orders in such a way as to try to guarantee that they're going to be able to deliver on their own promises downstream from themselves. And here's kind of my one big data slide that is supposed to illustrate what happens. So in this model, there's a lot going on. Let's focus on this upper left quadrant here right now. So here is over time. And just for simplicity, we're tracking across 4 tiers of the supply chain. So you should think of firm 0 as being direct to customer, firm 4 being very removed from the immediate customer, right? The customer of firm 4 is firm 3, and the customer of firm 3 is firm 2, and so forth. And what we see is there's 2 periods we put into this particular simulation. The flat parts are periods of relative stability. When there's no crisis, there are fluctuations, but it's kind of calm. And then there are these periods of shock. So in this slide, we included a demand shock where suddenly it either in reality or in perception, a lot of what firm 0 produces is needed. You can imagine this to be masking equipment, respirators, toilet paper, whatever you think is important in times of a pandemic. And what you see in the structural case, this is where the firms don't really they're either stuck in their contract, or they feel unable to adapt, or they don't think a real crisis is happening. The demand shock goes up. You see the blue spikes right up, and the blue's kind of hidden by the other curves here, unfortunately. But there's very little wobbliness in the blue, but by the time you get all the way up to level 4, the purple firm, the perception of demand that firm 4 experiences is all over the place. They have really high uncertainty. And the important thing, let's look in this second big shock here. It takes from about time 200 to time 350 or so for things to settle down for firm 4. So after a disruption, it takes a long time for them to recover and get back to their stable lives. Now, if we look over on the other side here in the policy setting, this is where the managerial agents in the system have more freedom to make decisions and alter policies in the moment based on the idea that a significant change is occurring. What happens is, so if you look in the periods of stability, there's more bullwhip effect. There's more just totally unfounded variation in perceived demand. But if you look in the time of crisis, there's still this big shock up and down. Purple is definitely the worst. Green firm tier 3 is also pretty bad, but firm 1 is experiencing much less, is recovering quicker from the shock. But the point is, if you look at firm 4, the far upstream firm, its disruptions about time 220 or so. Time 220 or so. But the recovery is by about 275. So the recovery is something like 3 times faster in the case of a policy situation where the manager is at each time point estimating what might be happening and trying to adapt. And so the take home of this in terms of policy is that there's a trade-off between these really stable approaches that maybe have long-term contracts and keep firms kind of locked into behavior, or bureaucracies or other procedures that keep people locked into behaviors. And in times of true stability, these can be really advantageous. But in times of crisis, there's the trade-off that you might not take the right steps early or even in the middle of the crisis to recover. And so this is illustrated in this in this diagram. And this is kind of the last big figure that I'll talk about. So, and of course, there's a whole spectrum of possibilities between a fully structured approach and a fully manager decision-making driven policy. And so, I mean, this is just one model. I certainly don't want to over-interpret it in terms of real life. Real life is vastly more complex than even this pretty complex model. But this trade-off between systems that lock managers in so they don't over-interpret problems that are not real problems, and the ability to react when an actual problem occurs without having to be too slow in their response time, is kind of the take-home message for non-math people from this model. Anyway, that's all. I'm happy to answer questions more. I'm aware I've taken up my time, but full credit to Tyler for exercising big leadership on this project. And I'm happy to be contacted by others who have questions kind of more at the PI level as well. So thank you.
Thank you so much. And, you know, given the insights we've heard in our first two talks, I'm sure this is going to be a really interesting Q&A session. These are very different topics, all very dynamic. So as a reminder to our audience, please feel free to drop the questions that you may have for any of our speakers in the chat today. And now I'd like to hand over the podium, so to speak, to our next speaker, Leila, who is going to be, oh, here we go, Leila Hedayatifar, who is a data scientist at the New England Complex Systems Institute, and she has actually also presented at a previous KIC webinar webinar in the past. And so we last heard about her research on quarantine policy, COVID modeling, and I believe it was November of 2020. So it's been a long overdue revision of her presentation. So Leila, I'd love to hand the floor over to you.
And you may have to unshare my screen. It's not really working for me. Sorry.
Okay.
Okay. Can I share my screen?
Yes, please. There you go.
Okay. 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 Hello, everyone, and it's my pleasure that I'm here in this webinar. Yes, the previous webinar was more than 1 year ago, and it was a result that we got for the first project that we did for an NSF granted project, and this is a new direction that we are going through it. And here I'm going to present the study and investigations that we did to check the effects of preventive protocols in COVID spread using an agent-based framework. Okay, and first I want to talk about why we study epidemic models. Epidemic models help us understand how infectious disease spread through a society, and it provides us insights about different factors that are crucial for developing effective public health interventions. And also it allows us to make predictions about the potential impact of a pandemic, and help assess the effectiveness of interventions like vaccination, social distancing, traveling restrictions, and other preventions. And also policymakers rely on epidemic models to formulate evidence-based policies and guidelines. And one of the most important thing is that sharing simplified epidemic models with the public can enhance understanding of disease transmission dynamics for people in public, something very simple that are understandable very easily. So based on these thoughts that we had in our mind, we developed a model in NetLogo. NetLogo is a multi-agent modeling environment and programming language designed for simulating complex systems and modeling agent-based frameworks. It allows you to have a figure for the whole model that you are developing, and the set up the parameters that you can consider different situations for them, like the slider or switch on and off icons for them, and also to have the plots and figures that show you how the model is evolving over time, and different compartment of the model is changing over time. I'm going to talk about these figures later. And now I try to go through different parts of the model we developed. Agent-based model, it specifies the dynamics by which a population of agents engage in interactions and undergo evolution through time. So we have a network of agents that they are connected to each other, and we consider different parameters and attributes for these agents or individuals in our network. First is the connections. And these connections represent that we have a network, we have a set of number of nodes or agents, and then we have the average node degree, which represent the connections for each node. So we connected the different nodes to each other based on that average number, and then we ask to relocate the nodes and put the nodes that they are connected to each other close to each other, which simulate the local connections for each person. And then over that, we create long distance connection that simulate if people travel to another location, or if they move different location and make connections with people who are not usually close to them. And then we consider demographic parameters like age and gender. For age and gender, we have the option that if we want to consider those effects in the model or not. And for the age, if we want to consider the age distribution, if we want to be a random uniform distribution, or if we want to use the US distribution for the population that got from the census. And also we are able to check the age range or the male and female percentage of population, which allow us to simulate different environments, which was one of the interesting things that we wanted to study. And the other one is the health risk levels. We have considered four different health risks: people who are older than 65 years old, pregnant women, cases that they have some health conditions, and cases that have serious health conditions. And it's possible for the model to define how many cases we want to have in our simulation and see what will happen in the model. And then we apply this IR model. It's a fundamental epidemiological model that is based on some differential equation used to understand and analyze the spread of the infectious disease. And it has three different compartments: individuals who are susceptible to the disease but have not yet been infected, individuals who are currently infected with the disease and are capable of spreading the disease to other people, and individuals who were infected with the disease and recovered and now immune to further infection for a period of time. And we have the parameters for them. First, the initial infected cases who guaranteed the start of the spread of the disease. And then we have the chance of getting COVID, which is the probability of transmission for the COVID. And it can be changed based on different variants of the COVID. We have the infected period of time and immune period of time. And the parameters that we have for the symptomatic cases and the symptomatic cases, when it starts, the active duration that they can transmit the disease, and distribution that we have for the symptomatic cases. And the one other parameter that was important for us was long COVID, that we want to be able to, if we want to study it or not. And it's just considered based on a threshold, that after that threshold, if they have still symptoms, they're going to consider them as long COVID. And a small portion of the cases that they were super immune, it's very small, but as it was reported in the model, in the data, we considered it in the model. Preventive protocols are the precaution ways that people help people to prevent the spread of a disease, like the ones that we had for COVID, like wearing masks, social distancing, or quarantine sick people, and also the vaccination. For these preventive models, we consider two different set of parameters: the precaution percentage that it relates to people, how they behave, and also the vaccination. And for vaccination, we consider that if we want to have the vaccination in the model or not, and when it starts, the percentage of people who are doing the vaccination, efficiency of the vaccination, decay in the vaccination efficiency, and also the vaccination priority that allow us to start the vaccination with people who are in high risk level. And the compartment model, in our modeling, these type of models allow us to agents to be in a specific status in each time, and we can track number of agents in each compartment over time. So if we click on the setup, then we're going to have a set of the nodes with the defined average connections and long distance connections and the other attributes that I explained in the previous slides. And then at each time point, the model check all the status of all the agents and update the status based on the previous status and the neighbors that they have. And then by running the model, we can see that how different compartments of the model is changing in the model over time. And also some other measurements that we can do. For instance, we were interested to see the number of reinfected agents in the model, how many times, how many nodes or how percentage of nodes are reinfected again, and also the productivity. And the productivity is calculated based on as symptomatic cases are not able to work professionally, they are not productive during the time that they are symptomatic. It's kind of, we are calculating the percentage of people who are having symptoms. And all of these things that I explained so far and the model in NetLogo allow a person to go through the model, play with the parameters, and see how different parameters, preventive policies can change the behavior of a system and how it's changed the situation and different compartments of the system. But if you want to see the average behavior of the system, we need to do the multiple realizations. And for this one, we need to make an ensemble of multiple realization with the same set of parameters that based on that, we can analyze the average behavior of the system and explore various potential outcomes of the model. Here, I'm going to just show you for a setup of parameters. When we run the model, we're going to see different situations happens because we have different randomness. We have some randomness in the model, and that randomness over time can change the behavior of the system. So we did some realization for any set of the parameters. Here, I'm going to show you just some of the results that we made based on our model. For each of these figures and for each point, we run the model for 100 times and did the average calculation for each of those ones. I have 4 figures here, and I try to explain these 4 figures in short time. The x-axis represents the probability of transmission, which is the chance of getting COVID. And then in the x-axis, we have average number of infections per node that shows that how many times they get infected. The color bar represents the average duration of the pandemic, that for how many times we run the code. And the stop time of the code is the time that we don't have any infected or immune case in the model anymore. So this graph represents that the average node degree as the beginning of the pandemic. If you remember that the policymaker tried to convince people to reduce their connections and try to minimize their connection and with other people. And here we show that by going from high degrees of connections to low degrees, you can see that even in high degrees of connections, even in low probabilities of transmissions, the disease is going to spread very fast in the system. And the average number of infections is around 1, which means that most of the cases get infected very fast in very short time. But as the connections reduces, we see that there is a shift for the higher probabilities that we don't see that the disease is going to spread fast. It takes time for the disease, and we have many reinfections for the cases. So some cases get reinfected and reinfected, and in very low degrees, you see that it's even for very high probability of transmissions, the runtime of the model is higher, and few cases they have reinfections. So it takes more time when we reduce the connection. It takes more time for the disease to be spread in the society and recirculate in it. And for the long distance connections, again, we see that when we go from 0% long distance connection to 20% long distance connection, how the behavior is changing. And you can see that, again, we have a change in the a transition for the probability of transmissions. When we go from 0% to 20% long distance connection, you see that when we have a lot of long distance connections traveling, even in short probabilities, the disease is going to spread fast in the society. But when we eliminate long distance, the disease takes more time and more time to be spread in the society, and not many cases get infected many times. And for the precaution, here right now, I showed the average duration in the y-axis. For precaution, we went from 0 to 100, and you see that when the precaution is 0, even with for low probability of transmission, we have a peak in the duration of the pandemic. It takes a long time. It circulates a long time in the society, and cases. All the cases get infected. But as we go to higher precaution, when like the ones that we see for 100 precaution, you see that even for high probability of transmission for the disease, not all the cases get infected, and the spread of the disease stops very soon. And this figure represents the results for the vaccination percentage. You can see that even with a small vaccination percentage that we do for the society, it we see the big change for the number of cases and the duration of the pandemic in the society. And for higher probabilities also, then the time it's changing, which is very important, because at the beginning, and remember that these vaccinations start just in the setup as a standard setting that we had. It started after 3 months. So we are also interested to see how it's going to happen for the other situation, but I didn't show it here. But you can see that even small changes can make a lot of differences. So in conclusion, I can say that our model depicts the spread of the virus within a population, considering a number of initial infected cases and whether it can propagate through the entire population or be contained before infecting some individuals. And based on those figures, I can say that reducing both local and long distance connections on average act as a deterrent to the rapid spread of the disease, and implementation of precautionary and vaccination policies not only decelerates the spread of the disease, but also lowers the likelihood of reinfection. These are some of the results for this work, and still is in progress. And we're going to have much more nice results in the future. And I want to acknowledge my colleagues in this work, and thank you for listening.
Thank you so much, Leila. That was really interesting. And I know that we're going to have some great Q&A questions as a result of your presentation. But before we get to that, I'm going to introduce our final speaker of the day, which is Deepika Ramasubramanian, who's here with her advisor, I believe, Leisha Palin, who's also here this afternoon. And they are both at, I believe, UC Boulder, correct? And are going to be presenting their research on disinformation and social media. I think, Deepika, you are presenting today, so I will turn it over to you.
All right, thank you so much. So I'm just going to do a quick slide check just to make sure you can see everything. Right. It's all good?
Looks good.
Okay. Okay, so good afternoon. Like Lauren said, my name is Deepika. Sorry. Okay, my name is Deepika Ramasubramanian. I'm a third-year PhD student at the Department of Information Science at Colorado Boulder. So we're going to talk about our research of Fertile Ground, Women's Bodies as Sites for Motivating Disinformation. This work was a collaborative effort between myself, Hande Patan, Taj Harris, Lindsay Diamond, and our advisor, Leisha Palin. So this work investigates the information disorder with respect to the COVID-19 vaccines in social media environments. In our case, that's going to be Twitter, as it happens upon targeted populations. And in our case, it's women and other people who menstruate. This work is a result of the NSF grant, the rise and propagation of anti-vax and anti-access social media campaigns targeted at disadvantaged and minority populations during the COVID-19 pandemic. Another piece of work that came out of that grant was the polyvocality of online COVID-19 vaccine narratives that invoke medical racism. So similar to our work that we're presenting today, this also examined information disorder on Twitter, but the targeted population were Black Americans. So if you're interested, that paper was published at CHI 2022. Okay, so before I get started, and I'm trying to do this in a narrative style, I need to talk about the data set that we had. So we have an in-house streaming collection service that ran from December 18, 2020 onwards. We also had to collect backwards through the Twitter API, the academic API, which no longer exists. But we went back to March 11th, 2020. We collected on vaccine terms, vaccines, and some variations of the word vaccine. And we were starting to see an emergence of infertility topics that were incorrectly making a connection between the COVID-19 vaccine and fertility concerns. So naturally, our first stage of research begged us to empirically identify in number and phases the COVID-19 vaccine discourse in relation to fertility concerns from pre-vaccine to post-vaccine availability. So to do this, we used a bottom-up qualitative coding approach so with a stratified sample from our data set. So we stratified 5% per month during this time. And we found 4 main constructs that were being talked about in those tweets that we analyzed. The first one is conspiratorial talk. And this is our classic conspiracies around vaccines and fertility. There is a depopulation agenda. Bill Gates is out to get the global South and sterilize women in the global South, and so on and so forth. The second kind we saw was flawed reasoning. And this was really an umbrella term for 3 other codes that we were seeing. The first one being bad science, where people used scientific jargon to give themselves a sense of credibility when they were making claims that were false. The second was cherry picking, where they would pick up one scientifically accurate empirical fact and then manipulate it to serve a nefarious purpose. In this case, in many cases, they were insinuating that the vaccine was causing cancer to people based on information in the vaccine insert. And then those that were making blanket statements, the vaccine causes infertility, with no real reasoning behind that. The third kind of construct was pro-vaccine arguments, and it was very heartening to see that move into the space, trying to quell some of that incorrect information on the platform. And finally, there were those that were uncertain about the vaccine safety because of the speed at which the vaccine was developed. The emergency use authorization was another cause for that genuine uncertainty. So these were the 4 constructs that we found, but we also found that they varied a lot through time. So after seeing how they varied with time and triangulating it with real world events, we saw that there were 3 phases of vaccine discourse that was really going on. So the first phase was when the vaccine was being spoken of theoretically. The second was when the vaccine was imminent. There were trials going on. They were starting to have some results from the trials as well. And then the third phase, when the COVID vaccine was widely distributed. So per phase, we saw conspiratorial talk extremely high alongside flawed reasoning. A lot of these arguments about the vaccines were really based on previous vaccines and the conspiracy theories and, poor reasoned arguments about older vaccines, especially the HPV. Now, in phase 2, as we start to get more information from the scientific world about the vaccine, flawed reasoning shoots up as people are trying to make false claims between the vaccine and fertility. In the final phase, we still see an extremely high flawed reasoning component, but at the same time, we're starting to see pro-vaccine voices enter the scene, trying to dispel some of the myths in that area. But what was most interesting to us was that in phase 3, we were starting to see people talking about disruptions to their menstrual cycles. So there were people self-reporting menstruation changes. But even further out than that, we saw people incorrectly associating changes to menstrual cycles to infertility. So for example, one person says, some people saying the vaccine is causing menstrual cycle changes in women and increasing rates of infertility and irregularity. Yeah, I'm good. No reasoning, but that has started to take root on the platform. So naturally, our next stage of research wanted to understand the range of positions, values, and arguments that were surrounding these self-reports and the menstrual disruption discussion. So to do this, we used discourse analysis because we wanted to capture the construction of the arguments and how people were interacting with those arguments through the response to tweets. So we also had to refine our data set to use tweets that had high convergent activity. And by that, we mean people that were using it to reply and using it as quote tweets, because really that's where the conversation was happening. So we constructed an engagement metric using those two metrics, which is replies and quotes, and then constructed a sample of 69 relevant tweets to perform discourse analysis on. And what we found was that people were primarily talking out of 4 rhetorical frames. The first one was the vaccine frame, and this was used to express people's opinions about the vaccine and usually involved a pro, anti, or basically a concerned stance towards the vaccine. The second was the menstruators frame, and this was used to describe menstrual changes, to acknowledge, deny, or question other people's experiences, to ask questions about menstrual health as it relates to the COVID vaccine, and also lament the negligence around menstrual health in the vaccine trials particularly. The third frame was the scientific, medical, and the public health frame, and this was used to ask questions to people in that authoritative position about the connection between the vaccine and menstrual disruptions. They were also expressing their opinions about the scientific, medical, and public health community. And others drew a sense of authority through their association with the scientific, medical, and public health communities. So the last frame was the lived experience frame, and this was used when people were reporting lived experiences of menstrual disruption or when people were validating or denying other people's lived experiences. So what we saw was the 69 tweets were using one or more of these frames in similar ways to make their arguments. So based on that, we found that there were 12 main perspectives, but I will only be touching on a few today in the interest of time. So the first perspective was using the platform to gather data about other people's menstruations, lived experiences. And I want to draw attention to Kate Clancy, who was the author of that tweet and also will be talked about somewhere further out in the presentation, where Kate Clancy was asking for people's lived experiences in a formal manner through a survey. The second type, we saw medical voices that were validating menstruators' experiences and providing expert advice or opinion about the phenomena. And here we have the user Vicky Loves Facts. Her name is Dr. Victoria Mail. We will also talk about her a little bit further down in the presentation. But here she uses her position as a reproductive immunologist to give her informed opinion about the connection between the vaccine and menstrual disruption. There were others who used their position as medical professionals to cast doubts on the vaccine. So this was like the flip side of something that we saw in the previous perspective, where people were incorrectly associating clotting disorders to menstruation and then onwards onto infertility. We have also those that were invoking scientific knowledge and expertise to dismiss lived experiences of menstruators so that they could protect compliance to the COVID-19 vaccine. So here, I want to again point out that the author is a very well-known Indian feminist, and she uses the position of authority of a celebrated gynecologist from India, saying that they verified that the COVID vaccine doesn't affect person's period, and please don't share that information online. And here, so we're seeing people who are interested in protecting the vaccine and compliance to the vaccine, but at the same time are starting to question women's lived experiences. And on the flip side, we have people with anti-vaccine stances that doubt the safety of the vaccine by demonstrating a new concern to women's health. So here's a quote here's the tweet by Candace Owens, who is, pointing out that the scientific medical community didn't have an explanation for the fact that people were having menstrual disruptions and that, we're worried. So we saw these perspectives, we analyzed their responses to see how people were engaging with various parts of the construction of arguments. And then we wanted to know, so what? So how is this propagating into the world? So our third stage of research wanted to understand how this information was being taken up and reused. So to do that, we did retweet analysis, and at the same time, we wanted to also understand how the word on Twitter was affecting popular press and information that was outside the Twitterverse. And to do that, we also did a popular press analysis, and I will talk about that right now. So for the retweet analysis, we picked up 2 tweets that explicitly requested people to share experiences of menstrual disruption with them, and then analyzed who retweeted responses to these tweets. So basically, who was retweeting people self-reporting menstrual changes? And we found out of the 192 self-reports that we analyzed that 176 of them were retweeted by at least one anti-vaxxer, and many of them were retweeted by quite a few more. We also found that popular anti-vax activist Naomi Wolf had systematically just retweeted 69 out of those 192. Another thing that we found were people were using that retweet aspect of Twitter to stitch together new narratives, recontextualize what the self-reports of menstrual disruption. So for example, here, they're using Naomi Wolf's commentary to give a new context to the self-reports of menstrual disruption. And how did this translate to popular press? We found that popular press was actually quite behind Twitter, and a lot of the incidents on Twitter were showing up in the popular press much later. So to do this, we did a popular press timeline analysis. We also analyzed each of the articles that we found between January 2021 to December 2022 to see if they actually had a stance on the vaccines affecting the menstrual cycles of women and other menstruators. So we found that this peak here in June of 2021 was a result of Kate Clancy's tweet that was asking for people's lived experiences of menstrual disruption. September of 2021 was Dr. Victoria Mayall's paper in the British Medical Journal asking for people to study the link between the vaccine and menstrual disruption. And then finally, the peak in July of 2022 is Kate Clancy's paper based on the survey responses that she received on Twitter was published at that time. But the other thing that I also want to draw attention to here is how few of these popular press articles were actually acknowledging that there was a connection between the vaccines and menstrual disruption. And even until August of 2022, not everyone had accepted that there was a connection between the vaccines and menstrual disruption, even though there was a controlled academic study in January of 2022 that was published that established a link between the vaccine and menstrual disruption. So what does this mean for us? We found that collective action online drew attention to an important medical issue that women and other menstruators were facing. We found that scientific and medical communities were silent on an issue for quite a bit of time, causing confusion and denial about the connection. An already mobilized anti-vaccine campaign, like we saw, mobilized in phase 1 before the vaccine was even created, was ready to incorporate this online reporting into their infertility claims and into their agenda. And then from an informatics perspective, we find that the best defense against disinformation is not allowing silence on the matters that affect marginalized populations, and that good information can do a lot better when it's placed as defense than when it is trying to correct bad information. that's all for today from me. Thank you.
Thank you so much, Deepika. That was fantastic. I want to conclude this series of presentations by thanking all of you for sharing your research with us today. And for the audience, let me say that each of our speakers' presentations will actually be available through our website and on our YouTube channel later this week. I'm also going to give a brief plug to our students who do a fantastic job of translating and transcribing all of these presentations into written English, French, and Spanish as part of our accessibility and multilingual resources initiatives. We actually just added French this semester, so that's a new foray where we're constantly learning what serves our audience best, and we're consistently adding new resources to our website. So I want to make sure to credit all of our students and our volunteers who help with this initiative. You can find copies of all of our previous lightning talks on our website via the link that I'm about to share with you all in the chat. But before we segue into the Q&A session of today's webinar, I also want to briefly plug the various ways that you can stay in touch with the COVID Info Commons. You can check out our website. We have a newsletter. Follow us on what is currently Twitter, Instagram, Slack, or LinkedIn. If you have any questions, you can reach us at info@covidinfocommons.net. And I will, of course, share all of these links in the chat. So peruse our channels at your own pace and keep an eye out for communications about our upcoming events and activities. But now I will stop with the administrative stuff and I will hand it over to Florence to continue our conversation this afternoon. We have several questions from our audience, so thank you for submitting those. But I'm going to give it to our executive director, Florence Hudson, to kick off our conversation today.
Thank you, Lauren. You always do such a great job organizing us. We're all very grateful. I bet I see a lot of smiling faces. So thank you very much for all of that. And thank you for these great presentations. It's always so inspiring and intriguing. And before I jump in, I just want to mention that 2 of our COVID Info Commons Student Paper Challenge winners are on the line with us today. So if they want to wave, we actually have Liam Cassidy here and Kiana Hendricks. So Kiana Hendricks. Liam is from Colby College and Kiana is from North Carolina A&T State University. So we're very proud of you and we're delighted that you submitted your research. So keep on researching and thank you very much. Okay, great. Now let's get into some of the questions. So I think we'll go back to the top with Gregory and try to make sure everybody gets at least one. Gregory, we have so many for yours though. Are you ready?
Sure.
So one of the things that I'm going to kind of scrunch a few of them together.
Okay.
As you were presenting and you were talking about potential positive impacts, of course, you start with mice, and I'm thinking, what about the humans? So do you think that there could be substantial potential positive impact in humans for the application of the research that you've done for long COVID with respect to brain fog or other impacts? And could the impact be positive if applied long after the COVID infection and perceived recovery? I know people that had COVID 2 years ago and were like, am I getting older or is this long COVID? Am I getting like new brain fog? Can this impact then? And to get it into humans, you probably have to do some clinical trials. So I was wondering if there's any plan for that. So those kind of all kind of go together. Like, how do we get this into the humans to help us?
Right. Well, I'll begin by saying, we do what we do in lab animals because we're trying to sort of mimic what we think is going on clinically. So that was one of the challenges early on. Some of you may recall, there were no good models, laboratory models of COVID.
Yeah.
So COVID had evolved to infect people, non-human primate, ferrets, hamsters, some other animals that aren't commonly used in labs. So that was sort of a big push for us. But to really answer the key question, absolutely. We think that by and large, it's now recognized that COVID is a vascular disease. When it first burst on the scene, we thought about the lungs, and of course, people were dying from pneumonia, acute respiratory distress syndrome. But once we sort of got past that, it's realized that it's affecting blood vessels. And really the disease is long COVID. And the science is still evolving. But there's clear evidence that every time one of us gets COVID, there's between a 10 and 30 to 40% chance that you develop novel medical conditions within a few months of your diagnosis. This is really borne out. And the symptoms often are completely unrelated to how you felt when you were initially ill. It's debatable whether getting COVID repeatedly increases your risks. I've seen data that suggests it does, and other studies suggest it really doesn't. But certainly prior to the vaccine, the incidence of long COVID was very high. But it's still, every time, I've had it a couple of times myself. I made it a good year before I finally went out of the house and, within a couple of weeks came down with it. So knock on wood, haven't experienced those long COVID symptoms yet. But yes, a lot of the things that I showed in mice, we've got evidence are occurring in the brain. There was a study done out of the United Kingdom, their National Healthcare Service, because it's centralized. They had, thousands of brain scans of people that were in good health prior to COVID. And then, several months to about a year after the pandemic started, they brought a lot of these people back and then re-imaged them and saw some worrisome changes in parts of the brain that we're seeing changes in our mice. And it's correlative, but a lot of these folks didn't have aggressive acute illness. They weren't all that sick. They were young. And they had, so-called hippocampal atrophy, parts of the brain that were smaller than they were just a year prior. So I'm not sure I want to see my brain scan, parenthetically.
I know.
Go ahead, I'm sorry.
Yeah, no, I was going to say, so we want to get to a few of the other questions. So I think your answer was,
Sorry.
yes, we think that this is going to work well in humans. And so we will look forward to when you tell us about how you can start doing those clinical trials in real live humans. And hopefully, I think we'll have forever to kind of figure this out. I hate to say that, but COVID's like around now, but the sooner you can get it out there, I think the better.
I appreciate that. Sorry, I could talk about this for hours. It's what
I could listen for hours. I know. I'm very interested. I think I want to talk to you again because I was thinking about lung to brain, cancer metastases. I'm like, oh, this sounds really similar, right? So I just think there are a lot of things in there that we can unpack. And one of my secret prayers is that if we figure out COVID, we figure out cancer, but that's another whole story. That's a dream of mine. So thank you. That was excellent and very intriguing. Now I'd like to go to a question from the audience, from Miranda, one of our other COVID researchers. A question for Leila. Wonderful work, yay. Can you talk about how you selected the size of the models and simulations, as in how many agents were used and how sensitive are results to the model size?
Okay, first, thanks for that question. That is one of the interesting questions for us as well. First, for these figures and plots that I show you, the setup was for 1,000 nodes. There was a slider for the controlling size of the network because we want to manage to have and be able to study different environments like a classroom, a wedding, or some ceremonies or a concert, something like that. So we are able to play with the size of the network. But for the sensitivity of the results to the model, that's something that we are still working on that. The first results that we got for the size is that we are able to find some scaling behavior for the other parameters, depending on the size, that when we are increasing and we're playing with the size of the network, we are able to find some scaling behavior. When I'm saying the scaling behavior, then it means that we don't really need to run the simulation for very large systems, because by the scaling behavior, we can scale the results and bridge to the point to define that what will happen for the larger simulations. Because when we go to bigger size, the running time is going to be much higher. It wasn't finalized. That was why that I didn't show it in this presentation.
Very interesting. Miranda, did you have anything else you wanted to ask on that? I think you're good. Okay, great. So now we have a question. Oh, thank you. Miranda says thank you. So we have a question for Zachary and a question for Deepika. Zachary, I hope you don't mind this question, but to make your analysis real to many of us, this might sound funny, was the toilet paper supply chain and demand challenge an example of this bullwhip effect? Or is there a different example that's more practical that you could share from the COVID pandemic?
Oh, that's a good, yeah, no, that's an interesting question. I actually don't know how things propagated up the toilet paper supply chain. I'll have to have that ready before the next time I give this talk, because that is a very real example where there was this momentary blip of demand, and then it kind of just went back to normal. That's a cartoon of what happened at the very least. As far as an example that maps kind of really closely onto this, I mean, yeah, I mean, one that's, I always am reluctant to opine on business issues because I'm not really a business person. I collaborate with business people and rely on their expertise. But my impression is that, like, there is some, it depends on what you think about AI and how you think global politics is going to go. But there's a possibility that we could be in a moment right now of semiconductor excitement. That is, I mean, we need semiconductors, but depending on how hyped the AI is and how mismatched that is with our actual ability to provide them, it is possible that this is one of those moments where, you know, the demand is going up, the perception of demand is going up a lot. I kind of think the AI thing is going to be real, but whether the hype is realer than is more than the real demand, I think that's certainly a possibility. That the perceived demand could go up astronomically when really it should have only doubled or something like that. You know?
That's interesting. And there's so many things to unpack in that question about AI and chips. And is it really different? And is it not? And what will the demand be? There's so many things, plus the market and people investing in companies and people going public and then IPOs. So there are a lot of impacts on that. Not as much in toilet paper, I don't think, but, you know, but thank you for bringing that up. That's very interesting. Very interesting.
Yeah. Well, now I need to look up the toilet paper example for next time.
I know, right? You know, I have a theory of why it happened, because I think that there were reports that in China there was an issue with COVID from that part of the body. And I think people read that and went, oh my gosh, it's going to come here. And then when it got here, it was like more respiratory, but I don't know. That was my impression, but I don't have any research analysis to support that.
Yeah. I mean, this is really well known in the bullwhip effect literature that once there's a fear that the bullwhip effect will create scarcity, then everyone's incentivized to exaggerate their needs even more and send an even worse signal up the chain, which is definitely exactly what happened with toilet paper.
Yeah, absolutely. A lot of very full baskets on the way out of Costco. Now to Deepika, very interesting research. There were a couple of questions that came in, so I just want to say them out loud and you decide if you want to do both or one, what order. Did you assess how uncertainty and fear could perhaps lead to flawed reasoning? It seemed the uncertainty, you know, on your bar chart was low, but I'm wondering about fear. And you know, maybe it's hard to ask people, are you afraid? You know, but I think fear can lead to a lot of, you know, flawed reasoning, right? Like, oh my gosh, I can't walk around that part of the city. It's like, it's fine. You know, so like fear, you know, can really impact. And then on the other side of affecting menstrual cycles, some of us might know that stress can affect your menstrual cycle. And when the world is in a pandemic, that's kind of stressful. So, you know, it could have been, I don't know if you did any, you know, correlation or causation related to the stress in the menstrual cycle, but I believe that's very real.
Okay, so I'm going to take the second one first, if that's okay. So this was the explanation provided by a lot of medical practitioners and other people, especially on social media. We saw a lot of people saying, oh yeah, but there's like a number of other things that can impact, including other vaccines. But I think it was so unprecedented that so many women, all and other menstruators, all at the same time got the vaccine. And then the next cycle gets impacted immediately after. So there are now academic studies from the obstetricians and gynecology people that say there is a connection between the vaccine and menstrual disruption. And there is, in fact, a mechanism through which this can happen. And so it's not really something that we thought, but it's more like, yes, it is true now. And it's known now that there is a connection. But at the time, no one was willing to admit that there might be a connection. And so it was so easy for, the anti-vax campaign to just pick that up and say, look, they don't care about you. Listen to us. It's going to affect your fertility next. So one uncertainty just bludgeoned into something, maybe another uncertainty, and then took off from there. And now I've forgotten the first question.
Okay, luckily it's written down. So we were talking about fear. Like you had uncertainty, and then you had flawed reasoning. And I was like, is fear in there somewhere? Because I think it could be fear. It may manifest differently, like other actions that people take, and you don't realize it's based on fear. It's something you can't see that they have, and they might not even realize they have it. So is there any implication of trying to look at the fear factor? So, actually, that has another meaning, but like fear as a factor?
I think, so what constituted flawed reasoning for us is, like, at the very least, there is something that they're trying to alarm the public with. Now, if someone was genuinely fearful and genuinely uncertain about the vaccine, it's less likely that they will be making statements, but would rather be asking questions. So the way we classified uncertainty about the vaccine includes fear about the vaccine. And I have some examples, but they're in my files, but we have examples in our forthcoming paper, hopefully, where we can show examples of, what the uncertainty looks like. And that looks a lot like fear. The flawed reasoning looks a lot like scaremongering, if I may use that word.
Oh, I see what you're saying. Well, when your paper is completed, please let us know. And if you all can update, your PI or co-PI profile, and you can add that in there, put it in your ORCID and give us your ORCID, then other people can find it too. That would be great, because thank goodness our funding goes for 2 more years. When I tell people, if you think COVID's over, my funding goes through 2025. But, there's a lot to still learn and apply here. And as all these boomerangs happen, and we get to keep getting it, and it keeps changing, and then a new one will come up. Not that I'm wishing that on anybody, but there's a range of uncertainty, as we say. I think it's going to be very important to learn from what you all have done and all the great and insightful work that you've done. We're actually thinking about, and I'm speaking to my publisher at Springer Nature. I've done a couple of books with them on cybersecurity work that I do. We're thinking about maybe inviting the PIs to be part of, a series of books. We're hoping to make it open access. So, years from now, when someone's trying to learn what all this great research said and what you learned and what your recommendations are for the future, that it'll be easy for people to find that. Because when NSF first called us and asked us to do this, I immediately went to the internet and said, okay, where's all the research out there? Very hard to find coronavirus research. There was a Springer book from the 1980s, from the second biannual coronavirus workshop. I'm like, they were doing this every 2 years and they didn't tell us anything, so I feel like it's our responsibility to document it well, in a longitudinal way. So we're looking at things like that too, so you all know. So hopefully this can benefit others in the future. So Lauren, I think all of our PIs answered the questions, and they were fantastic.
Of course they were. Yes. And I'm amazed that we got through all the questions in time, but that's perfect. It's meant to be. Thank you, everyone, and especially to our speakers and to our audience who joined us today. I don't know that we've scheduled our winter KIC webinar yet, but I expect that will happen in probably January. So we hope to see you all again. I can't believe I'm saying this, next year in 2024. And we hope that you'll join us for our next webinar. Please keep in touch. Please come to our upcoming working group sessions. Please keep an eye out for emails from us. We hope to keep working and collaborating with you all in the next couple of months.
Yes, and thank you for your great research and for being part of this community. Everyone be well, stay safe, stay warm, and stay healthy.
Thank you.
Thank you.